Hello buddies, welcome back to the channel Biopulse, and this is Chandrika. Today we are continuing the chapter that is sexual reproduction in flowering plants, and this is the lecture number 2. Let's get started. So in the last class we discussed about the male reproductive part, and today we are going to discuss about the female reproductive part. So when we talk about the female reproductive part, that is the gynoecium. Gynoecium is a female reproductive part of the flower, we can say, and it may consist of a single pistil or else many pistils, we can say. If the gynoecium is containing only one single pistil, then it is called as monocarpellary, and if it is having more than one pistil, that is 2, 3, 4, 5 many pistils, then it is said to be what? Multicarpellary. Mono, the word itself says single. Multi, the word itself says many. So here the word mono refers to single, and whereas multi, the word multi refers to many. And in multicarpellary, the pistil may be fused together, that is syncarpous, that is they may be fused. If there are many of the pistils, they means more than two pistils, if they are fused, then it is called as syncarpous, or else if they are free, then it is called as what? Apocarpous. See, for example, I gave here, this is the hibiscus pistil. Okay, this is the stigma, style, and then ovary. Only one stigma, style, and ovary is there. This is the best example for monocarpel because it is having only one carpel. And here, this is the stigma and this is the ovary. See here, ovary is fused. You can't differentiate them among themselves, isn't it? So that's what, if the ovary or else the pistil are fused, then it is called a syncarpous. Sync, the word itself says fused, and then carpus is pistil. Okay, and then here this is the multicarpellary syncarpous. Multicarpellary, why? Because many carpels are there. Syncarpous, why? Because they are fused of the papaver. This is the Papaveraceae family member, papaver. And this, see here you can see multicarpellary, many carpels are there. These all one unit is you are going to see, those all are said to be carpels, and they are apocarpous gynoecium because they are free. See, you can count them 1, 2, 3, 4 like that because they are the free. And this is the apocarpous gynoecium of what? Michelia. In exam, what happens? He used to give the diagram so that you must identify them. This is the hibiscus pistil, which is example for the monocarpellaries, and then this is example for multicarpellary syncarpous pistil of the papaver, and this is example for multicarpellary apocarpous gynoecium of the michelia. This is michelia, this is papaver, and this is hibiscus, papaver, and michelia. This is single one, that is mono. This is these two are multicarpellary, but this is syncarpous and this one is apocarpous. I hope this is clear to everyone. Next, each and every part of the pistil, when we talk, pistil has mainly three parts, that is the stigma, style, and then ovary. This is the stigma, which is said to be a landing platform for the pollen grain, where the pollen grains are going to be landed. And this is the hollow portion of the pistil, which is said to be a style, and there will be a bulged portion which is said to be ovary. Inside this ovary there will be a ovules. Okay, this is the landing platform, which one we can call it as a stigma. This is a style, hollow portion, and then bulged portion, the basal bulged portion is said to be ovary. And this is the pedicel, where upon which there will be arising of the ovary, style, and then stigma will be. Whenever the longitudinal section will take, you will be finding the anatomical structure like this. See, this is the stigma, this is the stylar canal, and these are the locus. Locus means nothing but it's a chamber of the ovule, where ovule is present. This is the ovule. This ovule is present inside this chamber. That's what that is said to be what? Locus. That, or else loci, you can also say many. Okay, inside the ovarian cavity, here there will be ovarian cavity. This all is filled with a cavity. That one is said to be ovarian cavity, which is also called as locule. The placenta is located inside the ovarian cavity. Where the placenta will be there? Placenta is the attachment, okay, to the ovary wall, to the ovule. This is the placenta, the bulged portion you can see. Arising from the placenta are megasporangia, that is commonly called as what? Ovule. See, if I zoom this one, see here, this is the placenta you can see. From this placenta, what happens? The ovule is bulging out. These ovules are also said that megasporangia. Okay, you can count them: 1 2 3 4 5 6. 1 2 3 4 5 6, isn't it? For each side there will be there is a fixed 6 ovules are there, or else you also call as them as a megasporangium. Commonly they are called as ovules. The number of ovules in an ovary may be 1, like in wheat, paddy, and then mango, to many, like papaya, that is papaya, watermelon, and orchids. If he is asking, give the examples of the mono ovules, means only one ovule is that, you can go with the wheat, paddy, and the mango. If you see mango fruit, one mango fruit will be having only one seed, isn't it? Because when it was young, during the flower of the mango, one flower will be having only one ovule. That's what the one fruit of the mango is having only one seed. Okay, ovary will form in future. Ovary used to go to form a fruit, whereas ovules, inside the ovary, whatever the ovules are there, no, they are going to form a seeds. To many seeds, if I am going to give you example, papaya you can give, watermelon, and then orchids. Next, this is the example and then structure of the megasporangium, ovule we can say, isn't it? This is the only one structure of ovule. Let me show you. See, if I say this is the ovary, okay, and inside this ovary, just imagine there is a only one ovule. Okay, so this portion is zomadia. So ovule is attached to the placenta by a stalk called funicle. Here there will be a placenta, here somewhere. This is the placenta, just imagine, and this placenta will be connecting to the ovarian wall, and this placenta, the ovule is attaching to the placenta with the help of what? Funicle. With the help of what? Funicle. This is the funicle, this one, this portion. Okay, next, the junction between the body of the ovule and then funicle is called hilum. This is very important. Whatever the junction, this one, this portion, okay, this is the body of the ovule. Overall this thing is said to be what? Body of the ovule. Whereas when the body of the ovule is attaching to the funicle, this is the funicle, isn't it? This one is the funicle, this one is funicle. Funicle portion and then body of the ovule is attaching, no, that portion is said to be what? Hilum. Okay, each ovule has one or two protective envelope which is said to be integuments. This one is one, and this one is second one. Two integuments are there, outer integument and then inner integument. Okay, this one is the outer one and this one is the inner one. Then opposite to the micropyle end there is the chalazal base. This is the base. See, this is the base. Okay, this one is the base. Opposite to what? Micropyle end, there is a opening which is said to be what? Micropyle end. Sorry, see, this is the micropyle end, sorry, micropyle opening. Just opposite to this, what happens? There will be a basal portion which is said to be what? Chalazal end. This is the chalazal. Okay, next there will be a, see these are the two integuments, right? There is a mass of cells which are called as what? Nucellus. This entire thing is called what? Nucellus. Inside this nucellus there will be a lots of reserve food material which will be providing nourishment to whom? The developing embryo sac. Okay, this is what actually the nucellus, inside the nucellus there will be a embryo sac which is actually the a female gametophyte. Okay, see here inside the nucellus, see this is the nucellus portion. Okay, this one entire thing is called as what? Nucellus, where it will be having, it contains the reserve food material which provides a nourishment to the developing embryo. Inside this one actually the embryo sac we can use to say, or else female gametophyte also you can say. An ovule generally has a single embryo sac. One ovule will be having one embryo sac. Okay, formed from a megaspore. From what? Megaspore. Okay, now let us consider the most important thing I can say, how actually the megasporogenesis is going to be happen. Likewise microsporogenesis we learnt in the last class, no? Same like that megasporogenesis is also there, but lots of difference is there among both of them. By the microsporogenesis we will get the male gametes, whereas by the megasporogenesis we will be getting the female gamete. So here it is the formation of megaspores from the megaspore mother cell, that is MMC. Already we discussed about this. Here MMC means megaspore mother cell. During the microsporogenesis MMC was what? PMC also we used to call, that is pollen grain mother cell. Here megaspore mother cell. Ovules generally differentiate a single MMC in the micropylar region of the nucellus. Just imagine now what happens, ovule is still young. Inside the still young ovule, inside the ovule, just imagine this is the ovule. Okay, inside the ovule what happens, there is only nucellus is there. At the micropylar end what happens, there will be a single MMC, that is MMC, megaspore mother cell, at the micropylar region of the nucellus, and it is a large cell containing very dense cytoplasm. Here in the picture you can see this is the only single megaspore mother cell, and this is having the dense nucleus and then dense cytoplasm also, isn't it? Why? Because it is going to be developed in the form of megaspore in the future, and it is the largest cell containing dense cytoplasm and then prominent nucleus. MMC undergoes meiosis to produce 4 megaspores. Okay, this will undergo, this MMC, it undergoes meiosis. Okay, this is the only one single MMC I can say, MMC, megaspore mother cell, which will undergo what? Meiosis. It is undergoing what? Meiosis to give 4 megaspores. There will be 4 megaspores. 1, 2, 3 and then 4. Next, in a majority of the flowering plants, one megaspore is functional while the others degenerate. Okay, one will be functional and other will be degenerate. They form there is a formation of 4 megaspore, isn't it? 1, 2, 3, 4. Among them what happens? 3 will degenerate, only one will become functional, and then other 3 will degenerate. All of these 3 will be degenerating. Okay, this is one MMC, that is megaspore mother cell, which will undergo meiosis to give what? 4 tetrad, same like that in the microsporogenesis. But here what happens? The 3 will degenerate, only 1 will become functional, and this functional megaspore develops into what? Female gametophyte. Okay, how it is going to be happen? That will be the next task for us. Female gametophyte is also called as what? Embryo sac. We used to call this one is also called as what? Embryo sac, or else female gametophyte. Embryo sac formation from the single megaspore is called as what? Monosporic development. See this one, this embryo sac is formed by only single MMC. That's what it is called as what? Monosporic development. Next, see, after the functional megaspore is formed, the nucleus of the functional megaspore, the nucleus of the functional megaspore divide mitotically to form 2 nuclei. They move to the opposite pole. See, from one functional megaspore, this is functional megaspore. Okay, this functional megaspore will undergo mitotic division. Okay, only nuclear division, nucleus is going to be divided. Here what happens? 2 nucleus is going to form, and the formed nucleus are moving opposite. Again second mitotic division. Okay, only nuclear division. Next, again in that opposite pole only what happens? One more mitotic divisions to form 2, 4 and 8 nucleated condition. Okay, here the nucleus is again divided 2 times, forming 4 nucleated and 8 nucleated stage of the embryo sac. How many times? Here the nucleus is again divided into 2 times. See, here one time and here second time. Actually this is first one, 2, 3. After the 3 mitotic nuclear division, we will be getting 8 nucleated stage. These divisions are free nuclear. Okay, then next nuclear division are not followed immediately by what? Cell wall formation. Only nucleus are divided. You see, isn't it? There is no formation of the cell. But after the formation of these nucleus, what happens? From the each pole, one nucleus move towards the center. From each pole, from each opposite pole, one of the nucleus will move towards the center, and remaining 3 will be remain same. That one we will discuss in the next slide. See, after the 8 nucleated stage, cell walls are laid down, leading to the organization of the typical embryo sac, or else female gametophyte. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. Remaining 2 nuclei are situated below the egg apparatus in the central cell. I will show you how they are going to be organized. See, for example, if I say there is only 1, 2, 3 and then 4 nuclei, isn't it? Next here also 1, 2, 3, 4 nuclei. From each pole up, what happens? One nuclei will move towards the center. From each pole, one nuclei will move towards the center. See here what happens? 1, 2, 3, 4 nuclei at the one pole. Another end 1, 2, 3, 4. From each side what happens? One nucleus will move towards the center and one more nucleus is moved towards the center. So that what happens? 3 nuclei will remain here and then 3 will remain here, but 2 will be at the center. Okay, that is called as polar nuclei. After that, what happens, there will be a form, after the movement of the nuclei, there will be a formation of wall around them, like this. So here there will be a formation of 7-celled 8-nucleated cell. This is what actually said to be what? 7-celled 8-nucleated embryo sac. Why it is called so? Because a typical embryo sac is 8-nucleated and 7-celled. See, for example, if I count 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus are there. But cell, if you count 1, 2, 3, 4, 5, 6, and then the large one is last one, that is 7. Okay, so that's what a typical embryo sac in the angiosperm is having 8 nucleated and 7 cell. 3 cells are grouped together at the micropyle end and constitute the egg apparatus. This consists of 2 synergids and 1 egg cell. Since this one is called what? Egg apparatus, whereas egg is there and then synergids will be there. These are the 2 synergids and this one is 1 egg cell. Female gamete it is. Okay, we found female gamete now, right? And this is the large one, that is central cell, we can say. Central cell which is having the 2 polar nuclei, and here at the chalazal end there will be 3 antipodals. At the micropylar end there will be a 3 group of cells which is called as egg apparatus, which consists of egg, 2 synergids, and at the chalazal end we will be having the 3 antipodals, and the central there will be a large big cell which is called as what? Central cell, and which is having what? Polar nuclei. This structure is very important for your theory purpose and for MCQ purpose also. And if you observe very keenly, in the synergids there will be a finger like projections, isn't it? These finger like projections are called as filiform apparatus. This is very important structure, I can say. Whenever the pollen tube is going to be enter here, no, whenever pollen tube is going to be enter here, means during the fertilization, the entry of pollen tube will takes place here, and there will be a much more help of this filiform apparatus, that they will guide the pollen tube inside the synergids. Okay, that thing you will come to know in the next slides. Just keep in mind, the embryo sac has 8 nucleus and then 7 cell. 1, 2, 3, 4, 5, 6, 7 and then 8 nucleus. Whereas 7 cell have 1, 2, 3, 4, 5, 6 and then 7 cells. Okay, I hope everybody is clear about that. Synergids have special cellular thickening at the micropyle tip called as what? Filiform apparatus. These are the filiform apparatus. They help the pollen tubes to guide into the synergids. 3 cells of the chalazal end are called as antipodals. Chalazal end will be having what? Antipodals, and micropyle end will be having the egg apparatus. Next, let's talk about the pollination, very important part of the chapter. It is the transfer of pollen grain from anther to the stigma of a pistil. Now, up to this we came to know how actually the formation of male gametophyte and then female gametophyte is going on, isn't it? After that formation, how actually the male gamete is going to be transferred to the female gametophyte. As you know, plant gamete or else plant itself can't walk, isn't it? If you imagine pollen grain, it won't have capacity of motility, isn't it? That's what there will be a transfer of pollen grain from one flower to the another flower, or else from the anther to the stigma with the help of pollinators. That process itself is said to be what? Pollination. It is defined as the transfer of pollen grain from anther to the stigma of a pistil. Depending upon the source of the pollen, pollination can be divided into 3 types: autogamy, geitonogamy, xenogamy. Autogamy means self-pollination, xenogamy means cross-pollination. In lightly I will explain here in this picture. See, if here the pollen grains will be there, if the pollen grain of the same flower is transferred to the stigma of the same flower, then it is called what? Autogamy. Auto means self. Geitonogamy means pollen grain of the flower is transferring to the pollen stigma of another flower of same plant. This is the same plant, isn't it? Same plant but flower is different. Next, xenogamy. If pollen grain is transferred from one flower to the another flower of different plant, this is one plant and then this one is totally different plant. So like this, three types of pollination is there. We will come to know in details here. Autogamy. Auto means self. Transfer of pollen grain from anther to the stigma of the same flower. Same flower is important here. See, this is the anther and this is the stigma and this is the one flower. If the pollen grain is transferring from this to this stigma, then that is said to be what? Self-pollination or else autogamy. In flowers with anther and then stigma, complete autogamy is very rare. Autogamy in such flowers requires synchrony in the pollen release and then stigma receptivity. What happens in such kind of flowers, that is complete autogamy is very rare, because if pollen grain is matured, stigma will be hang. If stigma is matured, pollen grain will be hang. There will be an accurate time in between these two. Then only what happens, autogamy is going to take place. Pollen reaches stigma of the flower, needs close anther and then stigma also. That is very right timing we can see here. This is the only one flower. Anther is going to be attaching to the stigma of the same flower. Then it is said to be what? Autogamy. That one is said to be autogamy. Next, plants like Viola, Oxalis and Commelina produce two types of the flowers. Like this is the Viola, this is Oxalis and then this one is Commelina. Here we gave the examples of two types of flower they are going to produce. What are the two types of the flower? This is chasmogamous flower and then this one is the clistogamous flower. If you see, clistogamous flower is never ever open, and whereas the chasmogamous flower is normal flower, whatever you can see. What is the difference among them? Chasmogamous flowers, which are similar to the flowers of the other species with the exposed anther and then stigma, like this, they will be exposed chasmogamous, whereas clistogamous flower, they do not open at all. In such condition, what happens, anther and stigma lie close to each other, so that when anther dehiscence in the flower, when anther opens, the pollen grains come in contact obviously to the stigma of the same flower, so that what happens, pollination will occur. Here what happens in the clistogamous flowers, where never the flower is going to be open, assured seed set will be there, assured pollination will be there, because there will be no escape of the pollen grain itself because of the closed environment, isn't it? So clistogamous flowers produce assured seed set even in the absence of the pollinators. This is the main thing actually. Clistogamous flowers, no need of the pollinators because they never open. Clistogamous leads to inbreed depression. Here what happens, due to the many types, again and again autogamy, inbreed depression will be there. There will be no formation of the new alleles, new genes, new characteristics. So this one is said to be what? Inbreeding depression. Next, in the geitonogamy, when we talk, this is the geitonogamy, transfer of pollen grain from anther to the stigma of another flower, of the same plant. See, this is the same plant. You can see in a single plant there is two flowers, but anther is transferring from one to the another, and they are in need of what? Pollinators. Okay, pollen move from different flower of the same plant, so via insects or else wind also you can say. They need what? Pollinators. But whereas autogamy plant won't need pollinators in such like clistogamous flowers. See here what happens. The geitonogamy is functionally cross pollination because it involves the pollinators, but genetically it is similar to the autogamy. Why it is genetically similar? Because the flowers are on the same plant. Okay, source of the pollen grain is also from the same plant. That's what. Next, the xenogamy, that is cross pollination, we can say. See, this is one plant and this one is one more plant. Here, transfer of pollen grain from anther to the stigma of a different plant. Transfer of the pollen grain from anther to the stigma of a different plant. It brings genetically different pollen grain to the stigma. So for it is called as what? Cross pollination. So up to this we discussed about the types of the pollination and then microsporogenesis. Let me take one more slide so that I can explain you firmly. See here, let me explain pollination in short. We have three types of pollination, right? Let me explain the types of pollination. First one, that is autogamy. Here what happens, single plant will be there which is having the flower. Anther of or else pollen grain of anther to the stigma of same plant, then it is called what? Autogamy, or else it is also called as self pollination commonly. When we talk about the second type, that is geitonogamy, there will be a plant, okay, and here is one flower and here one more flower. This is one flower and this one is one more flower. Anther to the stigma of the another flower of the same plant. Okay, then here is the third type, that is cross pollination. There will be two different plants. This is one plant which is having the flower, and one more plant. Transfer of pollen grain from one flower to the another flower of different plant, so that they need what? Pollinators. Okay, let me draw bee. See how beautiful my bee is, or else insect. Okay, so transfer of the pollen grain from one flower to the another flower of different plant, then it is called what? Cross pollination. Okay, so this is actually for today's class. For the next class we will discuss about the agent for pollinations. Okay, thank you one and all.